Yeah, thanks for the invitation, and sorry I could not come more this week. I would have liked to learn more from Jochen about all his different lectures, but I'm sure he introduced many things I will talk about already. And that's why I put weak seed banks so that we all agree on the definitions of the model so that there is no discussion about whether it's true or not. I'm interested in plants, and that's why we use the weak seed bank model, which is more realistic probably. But yeah, at the end, I'm happy to discuss about extensions to other models. So what I would like to show today is how to use coalescent results to try to do some different type of inference on seed bank and dormancy, and show you a couple of attempts we had, and some things which work and some things which don't really work, and maybe try to get at the end some discussion about what one can do. Okay, and yeah, so I'm not doing much of the work myself, so most of the things I will present is the work of Kevin and Thibaut, who are my PhD student. Thibaut now is a postdoc. Diala was a postdoc also in the lab working on selfing models. And we work also on multiple merger with Fabian Freund, and the selfing part was also done with some collaborators, so the two Stefan in Max Planck in Cologne. Okay, so I think I will skip many things very fast. I didn't know how much what you've done, but I guess there are many things you know already. So seed banks are important. I think you talked about it a lot. They are just a bunch of different results about seed bank and diversity storage, and the fact that it also affects selection and so on. I will not talk too much about this today. And some reviews from Jere and Jelen and Jochen. And I did one short one on plant, more focus on plant models. So what I would like to start with is just what are the tools we need. So I think you've done most of them, I guess, in the last days. I just highlight the main formula. So there is no theorem, so you can relax. It's a very easy talk this morning. So I will not prove any theorem, but just a couple of formula we will need. So the theta, the population mutation rate, which is for n mu. Okay, and I think all of you know the Wright-Fisher, the dual process in forward and backward to obtain also coalescent. And we will use the weak seed bank coalescent. I will show you in a minute. Okay, and what we also need, which maybe I'm not sure if you discuss so much about this, is we will need also recombination, because we work on plants, and we want to use also the effect of recombination along the genome to switch genealogy to do some inference. So that's something that if you work on bacteria and so on is a bit different, because you don't have meiotic recombination per se. And so here we will use results of the so-called ancestral recombination graph. And the main parameter is this effective recombination rate, which is called rho, which is 4n little r, which is the rate of recombination per site. Okay. And the, let's say, new advances which have been possible by combining coalescent and the ancestral recombination graph recently is to use full genome data. So here, let's assume we have 4 individuals that we sequence, okay? And we look along the genome. So if you follow, this is the genome length, you go along the genome and you look when there are mutations, right? Which are the stars. So there's some mutations that you see in the sequence data, okay? And what we want to do is along the genome, we want to reconstruct the genealogy of these 4 samples. Okay, that's the aim of this method. And how you do that, you can build it, you can build the local genealogy, and how you switch between genealogy is by an event of recombination. So an event of recombination will switch a branch of the genealogy to build a different tree further on in the genome, and so on and so forth. Okay, so this is what is called the sequential Markovian coalescent set of work, which was the first papers from Youth and Hein, who extended the ancestral recombination graph idea to few samples. And then there are a few papers, and the applications mainly is this paper from Lee and Durbin in 2011, the first one, who really proposed a method to do that. So there are 2 Markovian processes, right? So one is at a given site, when you reconstruct the genealogy, the coalescent, which is a coalescent process with the Markovian property in time. But you also have a Markovian process along the genome due to the recombination process. Okay, and so that means we assume that when you have a tree, you know you can get to the next one only by one event of recombination, which will change the tree, okay, and so on. But you don't need to know the other trees in a genome. Okay. So that's the main assumption of this sequential Markovian coalescent model. And if we zoom a little bit, I think a little bit out and look at what we know in population genetics. So this is usually the things that mathematicians don't want to consider. So life is very messy, right? It's very complicated. So when we do models, we focus on usually one thing, for example, dormancy. But of course, there are many aspects affecting the genome-wide diversity and the number of SNPs you see in a genome, and basically the mutation and the coalescent rates. Which can be life history traits. So dormancy is one of them, but you have also some other ecological life history traits. But you have also, for example, the generation times, and you have past demography, which affect the population size. If you do selfing or outcrossing, for example, which also will affect not only the population size, but also recombination rates. And this will affect, in return, link selection. So one of the main problems is, of course, that in real data, there is also selection, which we'll ignore most of the time because we want to do coalescent neutral models. But we should not forget that, of course, most of the diversity you see might be shaped also by effect of selection at some sites and the effect of link selection around the sites, right? Okay, and so I just like this quote because at least it justifies we do something useful, maybe, from time to time. Usually we used to laugh with Wolfgang Stefan, my postdoc supervisor, whether population genetics makes any sense itself. But okay, and so the two ratios which are in this graph, if we try to summarize this graph by two main parameters, in fact, you have this rho and theta again, right? Because this rho and theta will affect the recombination, it affects the link selection. It will affect, of course, it's based on the population size. Population size is a function of all this life history trait, right? And mutation is also in the theta. So these two parameters are in fact underlying this whole graph here. And what I want to highlight is that if you know these two parameters, you can do a lot of things with it. You can do a lot of inference, but it also allows you to maybe understand the real data. I think this is where probably the real power of population genetics has been now in the last 5 to 10 years using genome data to use this estimations. What is interesting is that in humans, actually, rho over theta is approximately 1. And it has kind of some consequences because many of the inference, when this ratio is 1, works really well. So the humans are actually super lucky because they develop methods to do inference where, in fact, they are in a perfect case where the method works because of this. Of course, they could not know before, right? But in a way, the problem is that this is nice, but most of the method developed for humans do not work for many species really well if this ratio is not close to 1. And I will show you some examples, especially if you have dormancy, that this ratio can be very different from 1, and that affects a lot the power of estimation. So the question I want to address actually in the talk is the other way around. So usually we try to estimate that we think we kind of know this ratio in humans, and we want to estimate, for example, demography, right? You want to know how humans have migrated all over the world, what's a change in population size when humans migrated to North America, or something. What I would like to do is now, if we think about non-human species, what about using this genome data to infer maybe the demography, but also this life history trait, right? So can we, from this ratio rho over theta, learn something about potential life history traits? And I think that's an interesting question if you work on plants, but other fungi, whatever you want. Of course, bacteria, to some extent, also is true, except that the rho is a bit different. I think you should be careful how you treat it. Okay, so the idea is that I will look at classic violation of the Wright-Fisher model, which underlies most of the human models of inference. And I will look at the seed bank and selfing, because we did this together. So these are just coupled in the model. And I will, at the end, talk about the sweepstake reproduction and large variant offspring production and some attempt we had recently. Okay, so just a little overview of how this SMC works. I didn't put the formula. If you want them, I can send you a PDF and where all of it is explained. The idea is basically you go along the genome and you look at the series of trees along the genome. So this is one tree, then will be the next one. And in fact, you kind of reconstruct a local RG, right? So you reconstruct a local ancestral recombination graph between these two trees. And what we look is the hidden states, X, which are the times to the MRCA of each of the tree. Okay, and we do that for pairs of sequences because it's too computationally expensive to do it for many samples. So the trick is to do it for pairs of samples. So you take only sample size n equal 2, and then you do all the pairwise comparison possible. And from all this estimation, you fill so-called a transition matrix. So what is the matrix? The matrix kind of, if you see the math, probably is obvious to you. I usually describe it for biologists in words. You take every 2 samples. So let's we take this sample here, and we look when was their common ancestor, right? And we cut the time into discrete intervals. So usually we cut it in 40 intervals. You can do less, you can do more, right? The more states you do, or the more intervals you do, the more resolution you have, but the more data you need. And if you take these 2 individuals, you say, okay, in this tree, their ancestor was in this time period 4, and then we look the same 2 in the sample, when was their coalescent time in the next tree, which has been modified by a recombination event. And if you see that if I take these 2 guys, you have to go up now to this time period 5. Okay, and then I look how many times I will count over all the trees in the genome. I count how many times I switch from one ancestor which was in the time period 4 to a time period 5, right? And then you do that for all pairwise. And this is here the little matrix which summarizes this. You have a hidden state of the tree blue, hidden state of the tree A by time period. Okay, and then I counted, this is just a number of count of how many transitions there are. Okay, so here you have 6 transitions because there are 4 samples and you take each time pairs, basically, and you get all these values. And then you do that over many, many trees, right, over the genome. So you get a matrix which is 40 by 40 with many observations in there. So we do it for 2 sequences. I'll show you how to extend it to more later. You have different algorithms. So this I will not talk. If you're interested, I can discuss later. Basically, these are your hidden states. These are the observed signal, which is the SNP. So you take 2 sequences, you compare whether they are the same or not. If you have a SNP, you code with 1. If they are different, sorry. If there is no SNP, it's a 0. So it's also computationally very efficient because you take any 2 pairs, transform into a 0, 1, and then you go along the genome and calculate what is the probability that if you have some SNPs, what is the size of the tree for this amount of SNPs that you observe. Okay, so this is the idea here. And how we do that, you have to calculate a transition probability, which tells you if I have a tree at one position, either there is no recombination, I keep the same tree, or there is a tree at one position and there is a recombination event, and this recombination event will swap, for example, this part of the branch to this part, and then the ancestor you see is now becoming more recent because you have a more recent common ancestor, or you have here within the same branch, or you allow this part of the branch to coalesce further in past due to recombination, and then you announce the time to the common ancestor. Okay, so this you can calculate the probability for each of this transition, and the probability depends on the recombination rate, which is little r. Okay, so you have to calculate a transition probability. Oh, yeah, I didn't mention. And you calculate an emission probability, which is a probability given the time to the most recent common ancestor that there are some mutations. Okay, so it's a Poisson process on the tree. And so this is what we do. Then you have a bunch of statistical tools afterwards that you can use to do the inference. So I will not go into details here because also that's not the most interesting part. We played around with a lot of things and changed different functions compared to what has been done before. You can sometimes improve the inference, sometimes not. Depends a bit on the amount of data and the resolution you want to get. So usually you get what is called the Baum-Welch algorithm, which is one of the classic used by most of the other methods. We developed also a full likelihood method based on this transition matrix. It's much slower to compute, but you get better resolution. It can be extremely slow if you have a huge matrix with a lot of data. So, okay. And so what does it mean? This is what we do. Typically, this is in a graph. I will show you many like this. So this is the time from present to the past. Okay, and this is here population size. And what we want to reconstruct here is the past demography, the change in population size in time. This is this black line. Okay, so the black line goes up and down because this is one of the hardest scenario to infer. Right? So the other ones are fairly obvious. You have to do them, but this is, I show that because when it works for this one, you can be confident it works for many difficult ones. And what we want is to exactly have in color, this is our inference, and you can see our inference is by time steps. Okay, so we have 40 time steps. These are 40 hidden states, and these are the one we estimate, and we want to match them as much as possible, this black curve. Okay. So here this is what we get as an output, and how we get it, this is here the transition matrix, which would correspond, and the more red, that means the more observation you have, and you can see that in this case, this graph corresponds to this scenario. The matrix is pretty full. There is a lot of observation everywhere, but the density of observation you cannot see because it's not fine grained enough, but the density of observation has the signal to recover the shape, right? Because the size of the tree varies, you will have different transition density in this matrix, and this is where you can estimate from the past demography. So this is based on simulation, right? We simulate, we know what we want, and we try to recover it. And then what we did here is just vary the amplitude, you can see, of the changes. So here we have a bigger amplitude, bigger and really big. So here this is a log scale, right? So it's a huge difference in population size. And this is the matrix which corresponds to each of this case. And you can see that the more the amplitude grows, you start having holes in the matrix. So here there is, the white means there is no observation. And we simulate a lot, and this is actually almost a theoretical one, because this is the best case convergence based on the expected theory of the model. And that means that in this case, you see the estimation is not very good, and there's just nothing you can do about it. At least with this method, it's not possible to get any better. Because there is no observation in the matrix, there are some transition between some size of tree which just doesn't happen. Right? And that's it. Okay, so that's a limitation, which is a theoretical limitation of the model, which was shown also theoretically in another way by Terhorst and Yun Song. And here, these are the limitations you have to be aware if you want to do inference, for example. Okay. So, and what is important is what we can get is we also get an observed transition matrix, which is based on our calculation, and the theoretical one based on the model. And if you look at the difference between these two, this is here, this graph here, you can see that the scale is very tiny, because what happens is basically, if you have a very good inference, so this is in this case, you should have almost no difference between the one you estimate and the theoretical one, right? And the errors should be randomly distributed in the matrix, okay? So I'm not sure if you can really see if it's random, but it's random. So now I will come back to this later, that's why I'm introducing this here. Okay, so what we wanted to do for the seed bank. So we first do a model which is based on this weak seed bank model. I will not go in this because this is basically, we've seen many of this already before. This is based on the Sky and Chronolascu paper, where they do the backward model here to have a coalescent with a time M window, which is the maximum time the seeds can stay dormant. Okay. When we use this coalescent model, you can compute the coalescent rate, which is scaled by this factor beta square, and this beta is basically a function of the germination rate. I think you've seen this before, right? So I don't need to do too much. What matters, I think, for, again, the rho over theta, that's the most important one, is that the theta is scaled by beta square, so by 1 over beta square. The recombination rate is also scaled by beta now, right? Because the recombination happens backward in time only in one lineage. So you have a probability beta to coalesce, to recombine backward in time. So what that means is that the rho over theta becomes beta times r over mu. And you can see that in humans, r over mu is approximately 1 because you have no beta. But now if your beta can have very small value, then you can get trouble, right? And also the recombination r over mu ratio can also drive this rho over theta to different value than 1. Okay. So there is no a priori value that this should be 1, right? And we see in plants, for example, in Arabidopsis, this is the r over mu is approximately 5, for example. Okay, so humans is just a very peculiar case. We can do the same for selfing. I will just need this formula later. For selfing, this has also been calculated by, there is a paper by Müller and Nordborg where computed this ratio. So again, they compute the theta, which depends on this rate sigma, which is the rate of selfing. So if sigma is 0, there is no selfing. If sigma is 1, you have very strong selfing, meaning one individual reproduces with itself. Okay, and so in plants, especially in Arabidopsis, that's very common. That's why we want to introduce it here. And again, you see the rho over theta depends on this parameter sigma. Now, okay, and yeah, so I've used the two together. This is the formula we will use afterwards in the analysis, where we have seed bank and selfing. So why not? You can do both, and you can just calculate this ratio. Okay, so what we do is now, I will go very quick here. We do exactly the same procedure as the SMC, but adapted for this ratio of emission probability, which depends on sigma and beta, if you have selfing and seed bank. You can do the same for the transition probability, which depends on the recombination rate. Okay. And we want to know, does it work? Right? So can we estimate anything? So this is already old stuff, so to say, from 2 years ago. What we show here is exactly the demography I showed you before. And we estimate here for different recombination, for different germination rates. This is the dormancy rate. So 1 means there is no seed bank, and 0.1 means there is already a fairly long seed bank, at least to some extent. Okay, so the lower beta, the longer is the dormancy. And what you can see is that if you change beta, the rho over theta changes, right, basically. And what we want to do is we want to not only estimate the demography, but also estimate beta. That was the aim. And you can see this is the estimation. This beta star is the estimate we get from the method. You can get a likelihood estimation of the best value. And I would say they are not too bad for biologists in general. It's not perfect. You always underestimate when it's 1. You cannot find really 1. It's very difficult. It gets close to 1, but you don't reach it. But for other value, we get pretty close. And the more data you have, the better is the estimation. So I think that was very encouraging. We did the same for selfing. So here we vary sigma from 0 to 0.9. You can see the sigma star is the estimate, and the demography is also well estimated. Always get a bit more noise with selfing in the recent times due to some effect of the coalescent we can discuss. What is interesting is that if you have both seed bank and selfing, you have a huge problem because there is non-identifiability. Because the whole method relies on estimating rho over theta. If you have a ratio of rho over theta driven by different value of sigma and beta, the same rho over theta will give you 2 possible different values of the parameters, and there is nothing you can do about it. Right? So this is not solvable. I think at least we did not find a way to do it. You will need other information, and I don't know what else you can use because this is full genome data, right? So there's not much else I can use here. So what we show is basically you cannot identify if you have no a priori, but if you fix some kind of priors, if you know whether there is seed bank or selfing and many species is already known, you can already shift your estimate into one part of the parameter space where it works. Right? So if you have a priori, it works fairly well. If you know nothing, yeah, the method cannot tell you much about it. Okay, we applied it to Arabidopsis. So the demography is totally boring. I mean, it's not very interesting. But what is interesting is that we estimated a sigma, which is around 0.86 or 87. And this was a little bit surprising to us because in the experiments, so people have done a lot of experiments in Arabidopsis, and they find a ratio which is around 0.95 to 0.99. So we were wondering, why do we have this discrepancy between our estimates and the real rate? So what we thought is, actually, what about if we now try to estimate a rate of seed bank or selfing, which changes in time? So now we assume that maybe there hasn't been the same rate of selfing forever, and maybe it has changed over time. Okay, so that's what we are doing now, and the paper is almost ready. So the intuition is that if you go along the sequence length of the genome, you look at your coalescent times, and what happens is that imagine you have a species which was before doing no selfing, so sigma is 0, and suddenly there was a change doing selfing, right? So this is the present, and that's the past. And basically what happens is that you have coalescent tree which might be relevant or which are due to the present time evolution in a selfing time. But then you have coalescent tree which will go up to far enough in the past that actually they are affecting by the selfing and by the outcrossing now. Okay, and it would be the same for seed bank. You could have no seed bank and then suddenly you have a strong seed bank, right? And your tree will change in time. So the part of the tree will just get, for example, lengthened by the seed bank a lot. So we did it here with selfing for a reason, is because also in Arabidopsis a lot is known. So it's easier to test that it makes sense. And so what you see is this effect of clustering of trees which are due to this change in time of the outcrossing. And what is interesting is that you can see that in the transition matrix. So we believe that there is something real there, because when you build a transition matrix, now, if you remember the ones I showed you, they were completely homogeneous. And now what we can see is that actually you have blocks in this transition matrix appearing, because you have transitions which are either between those trees in one part of the matrix, in the recent part, and then you have, for all the Eden state, transition between those trees. And you can see here you have like many trees which are just very old and transitioned by recombination with each other, because recombination rate is much higher in the past. Okay, so because the recombination rate is high here, you transition very fast between trees, and then at some point you go back again to one tree which was under the recent time with selfing. And this is what creates this nice signal in the transition matrix. And we thought, well, probably we should be able to do some estimates based on this. So we do the same thing. You just have to be a bit more complicated with the estimations. There we estimate the selfing rate. You can see this is constant, so we estimate constant. It's so good that you don't see the line, huh? So just believe me, it works. And here you can see one transition, then you have a bigger transition, and here you have a transition from selfing in the present, 90%, to completely outcrossing, no selfing in the past. And you can see the estimation is pretty good. So we estimate the time when it happens, and also the magnitude. Here we tried a bit more fancy graph with different amount of data to test the reliability. What you can do is also some gradual change, and also up and down. So maybe it was selfing, outcrossing, you can do all sorts of fun stuff. Again, in plants, usually we have only one transition, but in fungi and many other species, this might be, you have different transitions in different ways. Okay, and then we applied it to Arabidopsis, and what we find, this is here the estimate of the time, which are around almost 800,000 years ago. So we are pushing really back the method a little bit, because you reach the limit of coalescent times in this area. So it starts getting very difficult to get estimates. So you need a lot of data, but we are quite confident that it's true. And there were a lot of estimates from the molecular biology who also estimated at the same time. So that gives us confidence that the method is capturing something. So we are doing the method for the seed bank in time. It's a little bit more tricky because the variance in the tree gets more problematic. And so I think the resolution is not as nice. So that's why I didn't show the results. But in principle, the same should apply. But yeah, I think it's a more statistical issue of how many trees you need in the variance that needs to be looked at. Okay. I think we'll probably skip this, because I think I can come back to it. We developed a simulator for the weak seed bank, which allows us to simulate full genome. It sounds very stupid, but when you want to analyze data, you need this at some point to full genome. So this is just to show you that the simulator works. So this is there is no seed bank to 0.25 germination rate. The TMRCA increases, and that's basically showing you the intuition behind the recombination. This is here the linkage disequilibrium. So how linked are sites along the genome. And you can see that also with the seed bank, you have much less linkage disequilibrium than when you have no seed bank. It sounds pretty obvious, but actually, if you look at the math, but when you see it in real, at least it makes some sense. So with this, we look also at selection. And I just wanted to show you this for those of you who are interested maybe in looking at genome data one day in your life. One of the things is you can look at selection. So what we do here is have a selected site in the middle, and then we look what is the signature in terms of diversity around the selected site. So this model is called the selective sweep model, where you have very fast selection. Okay, the selection will fix immediately the allele very fast, and then afterwards you will have a time where diversity will be very low or close to zero, and then recover slowly in time by building new mutations which will appear. And this is called the hitchhiking effect and selective sweep model. And this is, of course, very important because we in biology are obsessed by Darwinian selection. So many people want to look at these genes in a genome. And what you do is you go along the genome doing some kind of windows, and you try to find when there is this drop of diversity, right? So you scan the genome by window, and then you see, oh, there diversity is very low. And then you start zooming in and say, is there selection? And what we show is actually this is without seed bank. So you can see that diversity is of course much lower because the there is less diversity in the species without seed bank. And this is with seed bank. And what is interesting is that the signal is much stronger. So you see this really big drop of diversity, but much narrower. So you can see that the width of this peak here, this inverse peak, is much smaller than this one. And this is because recombination is higher with the seed bank. So you have these two effects of strength of selection, but recombination. So in a way, it's good news and bad news. It's good news because it's narrow, meaning you have less genes and the portion of the genome will be small. The bad news is that, of course, if it's too narrow, it's very hard to detect in data, potentially. So because biologists wants to have statistics, right? So we use some method which are used to do it, and then we can do some power analysis. So this is without seed bank. This is the black line is the false positive rate based on different thresholds. So in this method, you calculate windows, you put a threshold value, and this is based on neutral simulation. Okay, and then what you want is to have as low possible false positive rate, which is here the number of sweeps you detect, so 100% to 0%. Ideally, you want this black line to be at 0, because you want no false positive. And you want the line here, which is, for example, just after the sweep, to be as high as possible to detect 100% of the sweep. Right? That's your ideal case. The problem is that, of course, it doesn't really work like this, and you have a lot of noise in the data. So sometimes you think this is a sweep, where in fact it's a neutral region that just by luck has less diversity. And that happens due to drift, and you have variance along the genome. So the whole trick and the whole game in this analysis is to fix a threshold which minimize the false positive rate and maximize the detection of real sweeps. Right? And this is where it gets more figure science than real science at that stage, because you have to just like play around with the simulation and play with what you believe might be true. And then usually what people do is when they do that, they just look at the best candidate and do some biology on it, right? Find a couple of genes. What we show in our case, this is with the seed bank. I think that's very encouraging. As you can see that the false discovery rate drops extremely fast to 0. So I think that's pretty cool, because that means that with the seed bank, at least you're very unlikely to pick up false positives. The problem is, of course, it could be that you might have problems to pick up all the candidates. Some of them you will not see. So nothing is for free in this world, right? So there is no free lunch. And so I would still argue overall that with seed bank is a good news, because you will decrease the false discovery rate. But yeah, the method is not perfect. We have to maybe adjust it a little bit also. Okay. So the last part I want to show you is some attempt we had with a different model, the beta coalescent, also with the idea to extend it potentially to strong seed bank, which also generate the same kind of signatures, right? So that was the idea. So this is work, yeah. So this is work from my two students, so Kevin and Thibault, and we work also with Fabian here for a while on this. Okay, I think skip all this because probably all of you know about sweepstake reproduction. In this case, we assume it's a neutral model. You have large variance in offspring production, right? So this happens maybe in fish, in crop pathogens. This is also very likely. You have this huge epidemic every year in the field. In the winter, you crush completely the population because the crops are gone, right? If you take wheat, an annual crop, it's removed. That means all the parasites have to go somewhere, and many of them die, and you have a huge bottleneck every winter and so on. There is also a lot of discussion about whether this happens, for example, in viruses. So Jeff Jensen and other people have done some analysis. Fabian worked also on tuberculosis, whether those would explain whether they're due to strong selective event or neutral model. I'm not sure we should, I mean, we can discuss about this later. I don't want to do it now, but at least there might be some signal of this strong and large variance offspring in offspring production. So just to maybe, if you never really looked at it, so you can have, this is the classic Kingman with the binary merger, and now we are interested in trees where you can have several merger in the same time. Okay, I mean a multiple merger, but only one merger at a time because it's a beta coalescent. Here this is the graph of an SFS. That's the classic signature. This is the signature under the Wright-Fisher model. This is a forward simulator we did here, and this is using the model of the Schweinsberg paper. And this is here a strong dormancy model that we had done to look quickly forward in time. And you can see that the SFS generates this huge U shape where you have an increase of the high frequency variant. So I'm sure Johan talked about this at some point, so I will not. Oh, you didn't? That's why you should have started this morning. Okay, so that's the signature that we look usually in the SFS, right? And so several people, including Fabian and Bjarki, Eldon, Jere, Koskela, were involved in trying to design statistics to look at this effect in the SFS. I like the SFS, I have nothing against it. The problem is that as a population geneticist, I have learned also that you have a lot of confounding effect in there because it's a statistics which summarize the whole genome data, right? So everything which happens in there, like selection, any local linkage, everything will affect the shape of this SFS. So I find it a little bit unsatisfying, and it doesn't diminish the paper by Jere and so on, but I discuss often with people to say, well, maybe we should do something else to use the full genome data rather than to stick to this summary statistics, which is very rough. One of the main problems is, of course, that the SFS is sensitive to past demography. So if you have very weird demography, it could generate potentially some at least weird SFS, and this you have to control for it, and that's very difficult. And any selection will also generate potentially this kind of SFS. So we thought, okay, let's try to do SMC because that's what we have learned to do now since several years. So let's try to adapt this for multiple merger. So one of the, okay, so we will use the beta coalescent model, which there are many papers about, and especially there is a paper by Johan and Matthias Birkner, which had predicted this effect. This is here now along the genome we simulated with our beta coalescent model. Linkage disequilibrium along the genome. So again, how linked are sites. And in the Kingman coalescent, you have, as I showed you before, a nice homogeneous decrease. And you can see here that this is not the case. This was predicted in this paper. Here we just did it with simulations to see what is the extent and how far we can push it. And you can see the problem is that the LD is increasing again. So that means that if I take sites which are far apart in a genome, they show strong linkage disequilibrium. And this is totally not expected under the classic Kingman coalescent. Right? So what is the problem? The problem is basically that this multiple merger, as highlighted by Matthias and Johan, is not Markovian along the genome by this property. Right? So that means that our SMC actually should not work, right? Because we do a Markovian property, we go along the genome, take a tree, calculate the property of the next tree, assuming a recombination event. But you cannot know when I'm here in the genome, I don't know what has happened before, I don't care. Right? And this property here tells me actually, if I'm here in the tree, maybe it's linked to this other tree far away. So how do we do that? So we thought a lot with Fabian about this for 2 years. We tried many things. I think we tried many statistics and thought, oh, can we jump between trees or whatever different things. At the end, we said, okay, let's just go back to basics and just do an SMC. You know, just start it. We know it will not work, but let's try it anyway. Okay. So our main idea is let's do an SMC, but now with 3 or 4 sequences. So we have to make more math to calculate all the transition probability. Right? So we said, if this is the true multiple merger model, merging, we want to distinguish this from a Kingman where you have the binary merger. Right? That's the idea. So you see that to do that, you cannot do pairwise, right? You need more sequences. So that's why we use 3. Then we did it also with 4, just to be sure that we are not missing anything. And again, we calculate, so we use the fact that we want to look this ratio rho over theta along the genome. Okay, and I think one of the main points is that we know how it works for demography, right? That's the advantage. We know our SMC, how it works already. So that gives us confidence whether we can recover something. Okay, so the first aim is to compute the emission and transition matrices or probability. Then we need this transition matrix for the beta coalescent using an SMC along the genome. The beta coalescent has a parameter ranging from 1 to 2. 1 is a very strong extreme case. The 2 is should be close to the Kingman coalescent. It's not, but it's should be close to it. So our method is called SM beta C, and we suspected there will be a problem, right? Because as I told you, we know it's probably non-Markovian. So Kevin, my student who wanted to do some deep learning, he said, okay, why not, in addition, doing some deep learning, because we can simulate, we know the trees, so we can do probably something with this. So what we thought is, we say, okay, we will use the graph neural network, because I think it hasn't been used in population genetics. The idea is that a coalescent tree is a graph, right? Basically, so we can just use this graph neural network and say, let's take a bunch of trees along the genome, give it to a machine learning algorithm, and train it to recognize differences between the models. Okay, so we have sample size 50, so we can use more sample, right? Because that's of course the advantage with this. We build trees, and we use 500 coalescent trees only. So I think it's not too many. We use only 5 coalescent trees to train the method. And then what we do, we train the method based on many demography and different parameters of this beta coalescent. Okay, and then we want the method to try to tell us, what is the value of this parameter and the demography at the same time. And then we can compare. So I think the advantage is that we did not want deep learning in a way to do something very complicated. We want to really know, can we compare the deep learning, what it's really understanding, knowing that we have already our SMC where we know the math, right? We know the method perfectly, and we only know its limitation there. So I think that's a nice, cute problem, because there we can work between the two and really understand where the power comes from. Or at least I find it more interesting than usual deep learning approaches, which are very rough. And so this is just to show you what is the problem. If you simulate here with parameter alpha, which is the parameter of the beta coalescent, so this is almost Kingman. This is very strong multiple merger coalescent with classic method. So these are methods which exist, which are similar to our first one we did. You can see that the demography is completely wrongly estimated, and you have a shift in the estimation of the population size, right? Which is not surprising, because you have this constant which scales the tree, and this method cannot know it, so they cannot understand it. And so of course they cannot tell you what is the real population size. Okay, so what are our methods doing? So these are the two methods we did. In red is the SMC approach, so this SM beta C, and the blue is the deep learning result. Okay, so these are the results we got 2 weeks ago. So they are, probably one could do a little bit better, but they are already based on many repetitions. So you can see when alpha is 1.9, close to 2, it works fairly well for both, which is reassuring, because the SMC approach should not be too biased, because you should not have this problem of non-Markovian property too much, right? But then the more you decrease alpha, you can see that the red gets off topic, right? Very often. And the blue still stays fairly reliable. And only when you start getting very strong, and 1.1 is even flatter, you see that both methods don't perform well. But this is, I think, not really due to the problem of the methods. This is due to the data, because Fabian has a theoretical paper where he shows that if alpha is too small, there is no information on the demography anyway, because all your coalescent tree only concern a very recent past, right? Because they're all completely crunched to the present. So actually, anyway, there is no information. So you can have the best method possible. There is no way around it. The multiple merger is removing the information. So I think that's not too bad that we see there is some effect, but I think probably there we can increase the amount of trees, but at some point we will not recover all the information. I think that's very likely. How good are we in estimate? So what is surprising is that our SMC method does not work really well in estimating the demography, but it actually estimates the alpha parameter pretty well. So this is the true alpha for different scenarios. So we vary a lot the demography here to make sure we don't get any bias. And you can see this is the estimated. So here we have 1.9, we estimate something close to 1.9. Here it's 1.7, 1.5, 1.3. So you see that we overestimated a little bit, because of course the SMC has this problem, right? We knew, but it's still not entirely bad. And this is with 3 sample size and 4. So you can see 4 is marginally better, but there's not much difference. So what that tells you is really that I think this non-Markovian property really kicks in when you get to low alpha. And this is what we were suspecting. And the only way, at least we found around it now, is to use a deep learning approach. And why? Because we take 500 trees randomly in a genome. And that's the trick you can do with the deep learning. You can choose trees. And of course, if you choose them randomly and make the algorithm learn are there dependency between them, it will just find out, oh, there are dependency in the nodes of the different trees, independently where they are from. And this is the constraint we release compared to the SMC, right? So in SMC, we have to take one after the next. So we have no choice. Okay. And just to, when we wanted to convince us, we look at the SMC. This is the matrix difference between the theoretical and the observed one or computed one we get. And you can see now that, I'm not sure if it's really clear, but you see that this matrix is not random. So the errors are not randomly distributed because of this non-Markovian property, right? Because you have these big blocks where there is obviously a huge linkage between hidden states that we don't account for in the method. Okay, so that it tells you maybe it's a bit sad because we do a lot of math, right, and a lot of statistics and don't get a method running because of this non-Markovian property. And we were, I mean, somehow we don't want to do only deep learning, right? So it would be nice to get something out of it. And actually, Thibaut and Kevin had a good idea. They said, well, now, if you think about the SMC, it's based on Markovian property, but why not estimating alpha along the genome? So for each tree, we estimate a different value of the beta coalescent. Okay, so whether it is more multimole merger-like or normal coalescent-like, Kingman coalescent. Okay, and if you do that, actually, it's the same as doing a method to find positive selection. Because what you're doing, you go back to the paper of Schweinsberg et Duret, which suspected or proposed that this multiple merger, they are exactly like selective sweep signatures. And what we do is exactly this. We go by window and we calculate for a bunch of trees with our SMC. We calculate the alpha, the beta coalescent parameter value per tree. And then when we do that along the genome, knowing that we are under a Kingman coalescent, we have this Markovian property. And what we see is exactly this. Here we simulated a selective sweep in the red part, okay, with different strengths. So these are very strong selective sweep and neutral. And you can see here we estimate the alpha value. And the more you go close to the strong selective sweep, you can see that the alpha gets very low, close to 1. And then when you have neutral, all the trees have alpha value which are close to 2. So in fact, by doing this mistake of doing a method which we knew would not work, we kind of turn it, which was a failure, so to say, but we turn it maybe now to our advantage by saying, actually, well, we have a method to detect strong selective sweep and multiple merger coalescent under a Kingman assumption of Markovian property along the genome. So in a way, it's like a double effect. He's cool, like in one way it works, but one way it works for something else. So that was kind of the last thing that we thought about now. And I think, yeah, we are trying to put all of this together and think with Fabian if there is something else we could do, but we have not found much alternative now to improve this method. Okay, and I will just do some conclusion. So yeah, what is important if you want to do the inference, if you use full genome data, is this rho over theta. And I think as soon as you want to use decent amount of data, this is the main parameter of interest or ratio of interest. Many methods are biased because they are based on human life history traits. So I think one should be extremely cautious when you apply this method for many species. And I hope that I convince you that at least if you have seed bank and selfing, you can use our method. It works fairly well. And we also think that we can estimate change in time, which I think is pretty cool and opens a lot of questions in biology about species which change, for example, selfing or dormancy in time. And we wanted to estimate the rate of multiple merger and demography. And I think at least what we can show convincingly is now that whatever the demography is, we can know whether there is multiple merger or not. And I think that's a huge improvement, in my opinion, compared to the SFS-based method, because there we are, I think, much more powerful to show that. But yeah, our SMC method has this problem to overcome the non-Markovian property, and we could not do it. But we showed that by very simple machine learning with graph, we can overcome this problem. But maybe there is more nice mathematics one could do. But we can use this method to detect selection along the genome, and I think that's really a nice tool also that hopefully people want to use. Okay, and with this, I will just thank again all the people who did all the work, so especially Kevin and Thibaut, who have done most of it, and Diala for the selfing, and Fabian, of course, for many discussions. Thanks for your attention.